And what I'm going to talk to you in the next 40 minutes or so is the emerging importance of proper RNA metabolism in neurodegeneration. So you can see immediately from this introductory slide that we are affected by a huge number of neurodegenerative diseases, but they occur at different stages in our life, but they occur in different areas of the brain. But one of the things a lot of these neurodegenerative diseases have in common is the accumulation, the aggregation of proteins in distinct brain regions. And so one of the major targets of research in neurodegeneration today is to understand why these aggregates are formed, what are the proteins that are contained in these aggregates, and what are the consequences of this aggregation. And once we will have an answer to all these questions, then we will probably have a proper and, I would say, functional therapeutic options for these diseases. Because, as you may well know, right, there is a huge problem at the moment, because unlike, for example, some forms of cancer, almost all these neurodegenerative diseases, there is no cure, right? Once you get them, it's just a question of time of when your higher faculties or your motor functions are going to give way, and eventually this will lead to death. Now, before we actually get into the topic, let me tell you why it is so difficult to study neurodegenerative diseases. First of all, because they are insidious. I show this slide because I think it's very visually impactful insofar that this is a patient affected by primary progressive aphasia, which is a form of dementia. You can see that by the time the diagnosis can be made by a neurologist, already a lot of the frontal temporal region is completely degenerated, right? So one of the major problems is that our brain, because it is so good at trying to repair damage, you start showing symptoms when you have already lost a huge amount of neurons. And this is incredibly causing a burden on the health system. So this is the situation in Europe that has a rapidly aging population, but the fact that you have a rapidly aging population in some way, fortunately, is occurring all over the world. So currently 16% of the European population is over 65. By 2030 it will be 25. And 7 million people in Europe will be affected by some kind of neurodegenerative disease. This is doubling every 20 years, and it is currently costing something like 130 billion euros per year to actually care for these people. Not to cure these people, but to care for these people. And this is a huge burden, so much that if you arguably think of what is the best health system in the world, the US, here is a map of number of people who actually are affected by Parkinson's disease, one of the most common neurodegenerative diseases, who haven't seen a neurologist in 5 years after the onset of disease. So there are areas even in the United States where diseased people simply have not been seen by a neurologist. So why do we get neurodegenerative diseases? And there are 3 factors that are always playing together. Of course, I will focus on the genetics, but first I will tell you a little bit about the age and environment. Because these 3 factors, sometimes one is more prominent than the other, they are all contributing to disease. So there are some genes, we'll see them in a moment what they are, which are very much involved in neurodegeneration. That is, if you have a mutation in these genes, you will almost certainly develop the disease. But then, if you look at most people affected by neurodegenerative diseases, whether they will get one disease or the other, they will get it rather early in life, we're still talking around the 5th to the 7th decade in life, which is still a 20-year span, so it's fairly long, or whether it will be fast progressive or slow progressive, then this will be decided by an interplay from all these three factors. So just a little question also to answer a question that was asked to Tito earlier on. Basically, why is aging involved in neurodegeneration? Well, the reason you can see here is that if you look at your proteostasis network, so the way your body is capable of renewing old proteins, of disposing of aggregate material or so, when you're young, this is very efficient. When you're old, this is very much inefficient. So the reason why you have these aggregates, a little bit of these aggregates, even in elderly, normal individuals, is that the brain, the cells as they age, they have an impaired ability to dispose of this harmful material. But there is also something more, because now with the ability of, for example, looking at specific RNA processing events in the brain of people that are associated with aging and of people who have who are elderly but are also affected by a disease, you will see that if there is some impairment in the alternative splicing, for example, profile of aging people, in diseased people, this impairment is way higher. So it's not that neurodegeneration is simply that as you get older, you are bound to get neurodegeneration. You will certainly have a loss in your cognitive abilities, but in order to develop disease, you have to have some disease-associated events that actually take place in your brain. What can you do about it? Well, there is a huge, and we won't talk about it at all, but I'll just give it to you to give you the big picture. There is a huge amount of research going on two aspects of protection. So one is neuroprotection, and this can be done, of course, by diet, exercise, and all the usual stuff that you can read in every magazine nowadays. There is a more medical associated approach that is neurorestoration, so where people are trying to develop stem cells that can be introduced in the brain and start replacing the lost neurons. And then, of course, there is the environment. Now, the environment is, of course, highly debated, all right? There is certainly a connection between the environment, because there are regions, for example, in Italy, where in the 20th century, people have been dumping all the waste from the factories in rivers, where the incidence of Alzheimer or Parkinson is 2 or 3 times higher than in other, let's say, less polluted areas. There are certainly, for example, in Parkinson, some, for example, chemical molecules like paraquat, which was used for a very long time as a wide-spectrum herbicide or as an insecticide and pesticide. And this molecule actually, when ingested in fairly high amounts, can give symptoms that are very, very similar to Parkinsonism, right? They can be recovered if you remove the exposition to the chemical, but you do have certainly an effect by the environment if it's highly polluted. And then, of course, there is another fact that, for example, if you look at traumatic brain injury, so this is actually an advice to all of you if you go on a motorcycle to wear your helmet, that the classical view was that you had if you had an accident, you could have traumatic brain injury, and this eventually, if something went wrong later on in life, could have led to Alzheimer's disease, Parkinson, or some types of dementia. But what is now clear from the analysis of the transcriptome of people who have had traumatic brain injury is that the changes induced by traumatic brain injury have a lot of elements in common with these diseases, right? So environment plays a role, aging plays a role, but what about genetic? And genetic is also a very much debated subject because this is a diagram showing how 20 years ago people were looking for genes that were correlated with neurodegeneration. And basically, you had to find a very unfortunate and large family where many people were affected by some form of the disease. You went through a genetic linkage analysis of all these people. You would have eventually narrowed down to a candidate gene, and then using, of course, validation analysis and co-segregation analysis in other patients, you would eventually establish whether that disease gene was associated with a specific form of neurodegeneration, or if it was just a random and unconnected gene in the process. So this approach is actually very effective, but is very effective only in genes that are strictly associated with neurodegeneration. So if you have a problem in these genes, you will almost always get the disease. And for this reason, starting from the, let's say, mid-90s to the 2000s, the analysis of these families only yielded just a few candidates. So for example, if we look here at amyotrophic lateral sclerosis, that is probably because of the ice bucket challenge, one of the most famous neurodegenerative diseases nowadays, you see that until 2001, this approach of looking in families where a lot of members developed ALS only yielded a few genes, mainly SOD1 and tau. Now, this has changed completely in recent years when people have started introducing the technique of high throughput of genome-wide association studies. So one of the big advantages in the technological breakthrough that have occurred in recent years, so our ability to sequence a genome at a very low cost and with quite good speed, is the fact that if you want to know which genes may be associated with a specific neurodegenerative disease, what you simply have to do is you take all the people that have that kind of disease, you take a control cohort of normal people, you sequence the DNA of all these people, and then you basically you go and look which single nucleotide polymorphisms are enriched in the disease population as opposed to the control cohort. So what you end up from a study of this kind is you see you have here all the chromosomes. In the dots, all the dots represent single nucleotide polymorphisms, and you can see that in the population that is affected by the disease, some of these single nucleotide polymorphisms are more represented. And the genes that are surrounding, that are in the vicinity of these SNPs, they are the genes that they can be kind of suspected to be associated with that particular disease. Of course, then you still have to go through the validation studies, but in this way, you can actually look at you don't have really to look at single families where the disease is highly genetically transmitted. And this has completely changed the field of neurodegeneration, because if you remember this slide, stopping at 2001, now in 2011, and now it's even bigger, you can see that the number of genes that have been found to be related, in this case still for ALS, has grown enormously. And this has allowed two things. So first of all, it has allowed to find what are the metabolic pathways, and I'll show them to you in a minute, that are associated with neurodegeneration. And also it has allowed to uncover a fairly interesting connection between cancer and neurodegeneration. Because one of the things that is becoming clear now that we are finding more and more genes associated with a particular neurodegenerative disease is that many of these genes are also involved with cancer. So what you think would be two completely different diseases, really what is turning out is that they are two sides of the same coin. In neurodegeneration, you have a neuronal loss due to a lower survival and an increase in cell death in the brain. In cancer, you have exactly the opposite. You have an excessive proliferation due to increased survival and lower cell death, but the genes that are involved in these two very different, you would expect, types of diseases are genes that are mostly connected with RNA binding proteins. So some RNA binding proteins, where they are mutated in different ways, of course, can give rise to neurodegeneration, and some others can give rise to cancer. And the two gray areas that now people are exploring, and this is what we are doing in ICGEB, is really trying to understand how to translate the importance of RNA binding proteins from the genes that are mutated in the disease with the disease spectrum itself. And the two areas, as I said, one have to do with RNA metabolism and the other with the autophagy proteasome pathway, but I will not be talking about this because we are in an RNA course. So what is the importance of actually finding out what are these RNA binding proteins? And this is actually clear from this scheme. So once I know if an RNA binding protein is involved in neurodegeneration, what I can use is I can use it as a biomarker. So I can use its expression to see if these people are likely to develop the disease or not in a period of time. So I don't have to wait for them to show the symptoms, which can occur much later when a lot of the damage has already been done. I can, of course, try to model the pathological features, and we'll see quite a lot about that during the lecture. Try to, of course, understand its function, to understand the basis why, when they are mutated or they are misexpressed, there is neurodegeneration. And I can also use them for screen for mutations. So I can identify people before they develop who are at risk of developing the disease, and I can use this information, for example, for clinical counseling, or just to, maybe try new preventive strategies that could be helpful. And just to show you what one can do for biomarker studies. So, for example, instead of looking at these proteins in the brain, because that's not always easy, our brain is not only the most complex organ in our body, is also the most protected. It's not easy to see inside your brain. So you can see whether these proteins are altered in the easily accessible tissues like the cerebrospinal fluid or the plasma, or even in just the blood lymphocytes. What I can do, of course, is to study what are the targets of these RNA binding proteins that are associated with disease, because then eventually I can find what could be the genes involved that give the neurodegeneration. And you can see here just an example of a study that we did 3 years ago on the protein that TDP43 that Tito was explaining to you this morning. And we did an NMR study with Frédéric Allen at the ETH in Zurich, where we crystallized the RNA binding domain of these proteins with a RNA target. And so now we know exactly what are the residues involved in this binding to the RNA, and also what RNA likes to bind. So you can use this information to search for genes which could be the target of this protein and which could be misexpressed during the disease. Again, you can try to model this in animal, like, for example, in this case, the best model we found so far is the fruit fly. So if you take this gene, TDP43, that is heavily involved in ALS, you remove its homologue in the fly, you see the normal fly has no problem moving in a petri dish, but the fly where this gene has been removed has a paralytic phenotype that is very similar to that of the disease. And so I'll show you how you can use this information to look for proteins, additional proteins that could be connected with this phenotype. And then, of course, you can study why this protein is aggregating in the brain of the patients. And this is just a summary of all the different possibilities that can lead to misaggregation, so the presence of mutations, an increased production of this protein, and Tito was telling you about this, or an impairment in the proteasome or autophagy system. Here there is the connection between RNA metabolism, RNA binding proteins, and the proteasome autophagy system, because if you have mutations at this level and you have an aggregate formation of these RNA binding proteins, then this aggregate will tend to become bigger and bigger and bigger and eventually cause damage because of loss of function or because the aggregates are toxic by themselves. And so once we have this information, we can actually try and model the aggregate formation both in vitro and computationally. But one of the things that, of course, because we are most interested in RNA, is try to understand what these proteins are doing at the level of RNA. And this is where the difficulties begin, because these proteins like to do many things. They are not just involved in splicing, editing, transport, stability of RNAs. They are involved in all of these processes, because if you remember the diagram that Tito showed you, as soon as the RNA is transcribed by the RNA polymerase, these proteins like to bind the RNA immediately, and they mediate all the processes, all the life cycle of this RNA from the beginning to the end. And as a tribute to the other speakers in this RNA course, I can tell you that there is not just HNRMPK that is found associated with long non-coding RNAs, but this TDP43 has also been recently described not just to affect the expression of some long-coding RNAs like MALAT1, but also to be affected by the expression of others. So the end point is that if you think, okay, so I found the gene that is associated with neurodegeneration, I can easily find what it does, what are its targets, and therefore I have solved the problem of neurodegeneration. The fact that these proteins are doing so many things means that whenever people do these high throughput studies to find what are the target genes, depending on the cell line where you do your study, or if you look at the patients, you see that there are many hundred of genes that change when you remove the expression of these proteins, but there is very little overlap between these different studies. And the reason why there is very little overlap between different studies is because these proteins, they not only like to bind specific RNAs, but they like to bind to other proteins of its kind, of their kind as well. So this is one of the reasons why they can be involved right from the beginning of transcription to the degradation of the mRNA, is because they don't just bind a single RNA and do one single thing. They bind the RNA, they bind a huge number of other proteins, and depending on which proteins they bind and which they don't bind, they will be able to mediate one aspect of the RNA life cycle or not. And this is very important for what could be the disease-associated events that I've just shown you in the previous slide. So just to give you an idea of the kind of experiments that we do to find out, we have some way using mini-gene systems of testing for the ability of these proteins to actually affect, for example, pre-Mrna splicing. You can see here, basically, this is a very simple splicing system where you have CFTR exon 9 being included in an mRNA transcript. This is an alternative exon. It can be included or skipped. If you have this protein, the one involved in ALS, in the aggregation in ALS, then exon 9 is not included in a lot of the transcript because this protein is binding near to the 3' splice site and is repressing the recognition of this exon by the splicing machinery. If you remove this protein, you can see that the inclusion of the exon 9 is very much improved. You put back a site-resistant version, wild-type version of this protein, you recover the inhibition. If you put back the C-terminus, where most of the protein-protein interactions are involved, you see that you cannot recover also the inhibition. And so this can be used, this system, to actually not only map the regions of the protein that is important for specific RNA processing events, but this information can also be used to try and understand what is this protein doing in events that may be linked specifically with disease. And in the last few years, we have actually concentrated on one of these events, and this is basically a brain receptor it's called SORT1. It's a brain receptor that has the task of bringing into neurons progranulin, that for neurons is an essential growth factor. And you can see that inside the pre-mRNA of this SORT1 gene, there is an exon. It's called a poison exon, and basically in humans, TDP43 is sitting just like the CFTR exon 9 example, is sitting near the 3' splice site and is actually inhibiting the inclusion of this exon. But what is quite interesting is that if you compare the sequence of this exon with the sequence of the mouse exon, they are almost identical. But the mouse exon is completely different as far as behavior is concerned, because in the mouse exon, TDP43 is still sitting here next to the 3' splice site, but if it is removed, the exon is included without any problem. And the reason why there is this difference between the human and the mouse, so in humans this exon is almost never included, is because there is this single nucleotide change inside this exon that in the mouse codes for an amino acid, but in the human it codes for a stop codon. So if this exon is included in humans, it gives rise to a toxic protein that is very harmful to neurons. If it is included in the mouse, it just gives rise to an isoform of the SORT1 gene that we know is perfectly functional. So why is this difference between human and mice? And the reason why is that together with Fateme in Mohegegi, a PhD student in our lab, what we have done is to map not just the binding site of TDP43 nearby to this exon, but also the binding of several other HNRMP proteins. And you can see here that, of course, because these exons are very similar, the picture is also quite similar, but there are some notable differences. So, for example, you have HNRMPA2 that is a splicing repressor that is just binding to the human exon, but is not binding to the mouse exons. And there are also a few other differences that could be important. And we tested experimentally the importance of this event this is in culture cells. So basically, the end result, because I don't really want to go into very much detail, is that in humans, if you remove each one of these HNRMPs on their own, you don't have inclusion of this exon. So this gives another idea that in humans, this toxic exon is very heavily repressed. But if you start removing TDP43, so the protein that is aggregating in the disease, together with some of the other HNRMPs, then the situation can change dramatically. And also in the human setting, you can have the inclusion of this poison exon. So one of the things that could explain why, if you have aggregation of TDP43, some people develop the disease early or some people develop the disease late, is because depending not just on the presence of aggregated TDP43, but depending on the relative expression of all these other HNRMPs, you could have inclusion, a big inclusion or a low inclusion of this poison exon. And this is exactly what happens, because if you take the brain of patients that are affected by frontotemporal dementia, that is a very similar neurodegenerative disease to ALS, just that you get dementia instead of motor neuron disease, but you still have the aggregates of TDP in the brain, you can see that if you, and you compare it to controls, you can see that a lot of the HNRMPs that are involved in this inhibitory effect in the patients, they are elevated. So it's a little bit like in patients when you have TDP43 aggregating as opposed to normal healthy people, the amount of these HNRMPs that help the repression of this exon is elevated in order to try and keep the inclusion of this poison exon as low as possible. So we decided to take this a step further, because of course you cannot go to all the TDP43 splicing events and actually repeat this kind of analysis because it's very long. So we decided to look at the wider picture and actually use flies to see which HNRMPs are contributing to TDP43 in the onset of disease. And the reason why we use the fly, well, I've just shown you. If you remove the homolog of this protein in the flies, this is the normal fly, the fly becomes paralytic. Then the other reason is because we already had a lot of information regarding this model thanks to our collaboration with Fabian Fegin here in ICGEB, and we knew that if we had these flies that are paralytic, these flies are paralytic because they have a huge amount of damage at the level of the neuromuscular junction. You can see this is a normal junction. This is the junction of a fly without the homolog of TDP43. But if you put back, instead of putting back the fly TDP43, you put back the human TDP43, you see that the architecture of the junction becomes almost normal, and also the flies recover a lot of the motility, and they also recover most of the lifespan. And the other reason why, another reason why we decided to use the fly as the perfect model to try and test these things is because this is actually, I think, probably the last experiment I did with my own hands in the lab. So basically, if you look at the fly TDP43 and the human TDP43, and you look at the proteins that they like to bind, you see that the human TDP43 likes to bind this set of HNRMP proteins in human that is very similar to this set of fly HNRMP proteins, and the opposite is true. So we know they actually can see each other very well. And finally, we also had some preliminary analysis where we showed that if you, this is a fly adult brain, if you remove TBPH, you saw all the defects that I've shown you in the movies, because you have some damage at the level of the brain development. If you removed some of these connected HNRMPs, fly HNRMPs, there was also some damage. But if you removed both TBPH and these HNRMPs, the damage simply became massive. So what we did was to actually, together with Fabian, to actually take the list of all the major RNA binding proteins that are expressed in the human brain, and you can see here an example, and actually we went to see how many of these proteins were conserved in flies. And you can see that there is quite a number of these proteins that are perfectly or quite conserved in flies, and we had this opportunity of actually making a set of transgenic flies where the expression of these proteins was abolished. And so what we planned, and what we did actually, was to actually take these flies where each one of these proteins was, the expression of each one of these proteins was silenced by siRNA, and we crossed it with the flies that were either expressing very little amount of TDP43 or 50% of the amount of TDP43. Just to express it in two different backgrounds. And you can see that what we managed to get is a picture very similar to the one we saw for HNRMPA1. So basically, you can divide all the other HNRMP proteins in two different classes. In this first class, these HNRMP proteins are very efficient in further impairing the locomotory behavior of the flies when they are crossed to flies that express only 1 or 2% of TDP43, but they also can do that in the flies that express 50% of TDP43. But then also there is another class of more powerful HNRMPs, right, that are very effective with the flies that express 1% of TDP43, but they're not effective when you have 50% of TDP43. So the end result is that if you have a damage at the level of a specific RNA binding protein, either because it's aggregated, it's mutated, or whatever, and you remove any of the other HNRMPs expressed in the brain, then very likely the amount of neurodegeneration that you're going to get is going to be very much enhanced. And also, there is another interesting result that we managed to get from this study, because we also try to overexpress TDP43. So you cannot do that in the fly because it's lethal, but you can overexpress it in the eye. So if this is a normal fly eye, you overexpress TDP43, you see you have a very much an increase in neurodegeneration, because as Tito said, TDP43 has to be autoregulated, because if you have too little, it's going to lead to neurodegeneration. If you have too much, it's going to do exactly the same thing. And so we had these flies that were overexpressing TDP43 in the eye, and we crossed these flies with the flies where the expression of all the other HNRMPs was selectively silenced. And the idea was to go and see whether the neurodegeneration was getting better or was getting worse. And the end result of this is a little bit more varied than the loss of function experiments. So what we managed to find is that you have a set of HNRMPs where if you remove their expression on top of the TDP43 overexpression, the neurodegeneration is increasing. But you also have 4 HNRMPs, for example, shown here, where if you remove their expression in the presence of TDP43 overexpression, the phenotype of the fly eye is actually restored. So removing these HNRMPs has a beneficial effect on the overexpression mediated by TDP43. And once you have this result and you go to present this data, then of course reviewers will always ask you, well, but you know, this is fine, you managed to cure TDP43 overexpression in the fly eye, but what about humans? Humans are not flies. And so what we have started doing in the lab now with Sara Cappelli and Filippo Witt is to actually see if these results can be translated to the human neurodegenerative setting. And what we have done is to take a few alternative splicing events that we know from a number of previous studies, some from our lab, that are affected by the absence of TDP43, and we have tried to see what happens if you remove from these eukaryotic cells the homologs of the fly HNRMPs that seem to be protective. And you can see here is that in some cases, in many cases, what happens is that if you have this gene, for example, TNIC, that is a neuronal specific gene that nobody really knows what it does, except for the fact that it's highly and only expressed in neurons. Basically, you can see that you have this splicing event that in the presence of TDP43 is completely repressed. You remove TDP43 expression by siRNA, and you see that you have a big increase in the inclusion of this exon. But if still in the absence of TDP43, you remove HNRMPQ, that is one of the 4 HNRMPs that were found to be protected, you see that your splicing profile goes back to exactly the normal situation. So this, what this is telling you is that this is true also for several other examples, that I don't want to go into detail. But the conclusion from all this is that you can actually translate directly your results from the fly to the humans. And so now we have a tool to actually understand how TDP-43 not only can cause neurodegeneration by altering specifically some alternative splicing events, but we also have some clues on how to modulate the expression of other HNRMPs in order to recover the normal splicing profile. And I should tell you that this is not just limited to splicing, because we can do exactly the same with regards to gene expression. So, for example, in this gene, this is a gene that is also important for neuronal survival because MAD is a apoptotic factor. Basically, if you remove TDP-43, you have an alteration in the gene expression with respect to normal cells. But if you, on top of this removal, you also remove one of those protective HNRMPs that I've shown you, then you can recover the same gene expression profiles as in normal cells. And what is now, we are now trying to understand is why this occurs. Of course, there are several different possibilities. You could have that either these HNRMPs can affect the expression of TDP-43 itself. You can have that these HNRMPs that can bind to the TDP-43 protein and regulate its functions in some ways that we still don't know. Or also that these HNRMPs really are not directly connected with TDP-43, but they are binding to the same transcript and are just having the opposite effect. So if TDP-43 is promoting splicing, they inhibit splicing, and vice versa, or gene expression. We have started testing this, and basically you can see here that we already ruled out that removing these HNRMPs can have an effect on TDP-43 expression, because if you remove 3 of those out of those 4 HNRMPs, you see that the levels of TDP-43 remain exactly the same as the wild type. And also, we also ruled out the fact that if you remove TDP-43, the gene expression, the level of expression of these proteins is also affected. So this is already, this possibility I think already has been ruled out, and the people in the lab are now testing whether these HNRMPs can bind directly to TDP-43 and whether they can bind the same transcript. So I think I will actually stop here, but basically the conclusions I wanted to tell you is that if you manage to find which genes are responsible for neurodegeneration, and a lot of these genes have to do with RNA metabolism, you can actually find not just why this protein is aggregated, under what conditions this protein is aggregating, and what are the consequences on the levels of RNA expression, so both alternative splicing or gene expression. But you can also see if other proteins are capable of modulating this damage, so that this increases the amount of potential therapeutic targets in order to fight the consequences of aberrant TDP-43 functions. And also, what is important is the fact that if TDP-43, let me go back, if TDP-43 is very difficult to act upon, because as Tito told you, it is an essential protein that if you overexpress too much or downregulate too much can be harmful. So it is a very difficult protein to target from a therapeutic point of view. Maybe targeting these other HNRMPs could prove to be a better option in the future. And I'll leave you with this, and I'll take any questions you have.